Chapter 9. Homology and its possible causes. Science deals with the empirical, with what we can see, touch, and measure. But Darwinism goes way beyond that, making claims that have not even remotely been observed. Indefinite departure from the original type is perhaps the best example. What the evidence shows us is not indefinite departure, but oscillation about a mean. With homology, on the other hand, we come to something that is really observed: the shared characters of organs all across the animal kingdoms. Humans, for example, resemble dogs, monkeys, and other mammals in having hair and other mammalian characters, and we resemble mammals in numerous other respects. At a more general level of classification, that of vertebrates, our limbs share bone structures, and so do our digits. There are usually 5 digits on each limb. We also share 2 eyes, 2 ears, and so on. Homologous organs that are shared by different groups are far too numerous to list. The underlying similarity between groups is not superficial either. The homologous forelimbs of bats, horses, porpoises, and humans constitute one of the best-known icons in biology. They illustrate the artistic idea of variations on a theme. It is impossible to look at their structure without acknowledging that homology is a fact. In Evolution, a Theory in Crisis, Michael Denton quoted the 1980 Encyclopedia Britannica as giving the pride of place to homology in discussing the evidence for evolution. The indirect evidence for evolution is based primarily on the significance of similarities found in different organisms, the Britannica wrote. The similarity of plan is easily explicable if all descended with modification from a common ancestor. If, please note. More recently, there has been a tendency to substitute because for if. More cautiously, Wikipedia says that evolutionary theory explains the existence of homologous structures adapted to different purposes as the result of descent with modification from a common ancestor. Homology was well known to Darwin, who wrote in Chapter 13 of The Origin, "We have seen that the members of the same class, independently of their habits of life, resemble each other in the general plan of their organization. This resemblance is often expressed by the term unity of type, or by saying that the several parts and organs in the different species of the class are homologous. What can be more curious than that? The hand of a man formed for grasping, that of a mole for digging, the leg of the horse, the paddle of the porpoise, and the wing of the bat should all be constructed on the same pattern and should include the same bones in the same relative positions." How did it happen? On the ordinary view of the independent creation of each being, Darwin went on, it has pleased the Creator to construct each animal and plant in just that way. But Darwin thought that homology was better explained by appealing to common descent, to the existence of an ancient progenitor which embodied the underlying character. He continued, "If we suppose that the ancient progenitor, the archetype as it may be called, of all mammals had its limbs constructed on the existing general pattern for whatever purpose they served, we can at once perceive the plain signification of the homologous construction of the limbs throughout the whole class." So heredity explained the similarity, and that was one part of Darwin's explanation. The other was natural selection, the selection of slight modifications, each one profitable to the modified form. That explained the difference between the homologous organs of different species, the difference between the forelimbs of bats and porpoises, for example. By such means, Darwin argued, the bones of a limb might be shortened and widened to any extent and become gradually enveloped in thick membrane so as to serve as a fin, or a webbed foot might have all its bones, or certain bones, lengthened to any extent, and the membrane connecting them increased to any extent so as to serve as a wing. Yet in all this great amount of modification, there will be no tendency to alter the framework of bones or the relative connection of the several parts." Without further evidence or argument, Darwin asserted that in changes of this nature, there will be little or no tendency to modify the original pattern or to transpose parts. But why not? Natural selection could transform anything on his theory. One might expect that it could as easily make fundamental changes in organs as it could preserve them in outline. In fact, one expects exactly that, if natural selection is to explain the transmutation of species. But Darwin had found a formula that suited him. It could account for both underlying similarities between species and the differences between them. It could preserve the theme while introducing new variations. In fact, he could make it come out any way he wanted. A bone needed to be thickened or lengthened, connecting membranes increased, but without modifying the original pattern. Coming right up, sir. Natural selection at your disposal. To many of his contemporaries, that seemed like good science, and his argument that common ancestry and natural selection explain homology is widely accepted today. So it is time to ask, has our more recent knowledge of DNA, mutations, and embryology shed any further light? The whole topic needs a closer look. Homology before Darwin. Homology was widely recognized before Darwin. The comparative anatomist Richard Owen used the term in the 1840s. The founder of the Natural History Museum in London, Owen was the leading critic of Darwinism in his day. He identified homologous parts as the same organ in different animals under every variety of form or function. Owen also distinguished between homology and analogy. The wings of bats and birds, for example, are analogous, but not homologous. Both aid flight, but they are built quite differently. Feathers don't resemble the skin flaps that propel bats through the air, for example. Their design is quite different. Furthermore, some characters are shared by many species but are admittedly not present in a common ancestor. The example most frequently given is that of human and octopus eyes. Those eyes are structured somewhat similarly, but on any understanding of the evolutionary tree, they are remote from one another. Evolutionists have labeled such structures as homoplaces, but the label does not remove the difficulty that they pose for Darwin's theory, that such complex and similar structures could have come about separately at widely different times by chance mutations would strain the credibility of Darwinism. In fact, the phenomenon of homoplasy closely resembles the phenomenon of convergence, which has related problems, discussed in Chapter 10 below. After Darwin argued that homology could be attributed to a common ancestor, subsequent evolutionists took charge of the idea and went to work. In its neo-Darwinian reincarnation, homologous characters are defined as ones that are inherited from a common ancestor. Ernst Mayr proclaimed in The Growth in Biological Thought, 1982, that the biologically most meaningful definition of homology is this: a feature in two or more taxa is homologous when it is derived from the same or a corresponding feature of their common ancestor. In The Flamingo's Smile, Stephen Jay Gould followed Mayr down the same path. We call parts of two organisms homologous when they represent the same structure by a criterion of evolutionary descent from a common ancestor. No concept is more important in unraveling the pathways of evolution, for homologies record genealogy, and false conclusions about homology invariably lead to incorrect evolutionary trees. Ernst Mayr reiterated his earlier position in his last book, What Evolution Is. He claimed that Darwin said that certain characteristics of two species were homologous if they were derived by evolution from an equivalent characteristic in the nearest common ancestor of the two species. But in 2009, Richard Dawkins, of all people, rebuked his late colleagues, although not by name, for this faux pas. Gould died in 2002, Mayr in 2005. If we want to use homology as evidence for the fact of evolution, we can't use evolution to define it, he wrote. We question Dawkins' phrase, the fact of, but that is a separate issue. Otherwise, he was on target. He saw that what Darwin had proposed as an explanation for homology had more recently become its definition. One might even say that homology itself had evolved under Mayer's tutelage. Mayer quietly assumed that common ancestry is something that can be ascertained independently of morphology, as though we already have at our fingertips a vast family tree of branching varieties and species. If that were so, we could simply look up common ancestors and determine which groups share homologous features. With Darwin, evolution is still a theory, and homology is part of its evidence. With Mayer, the assumption is that evolution has been independently established. The controversy has raged ever since William Dembski and Jonathan Wells wrote in The Design of Life, adding, "Neo-Darwinists continue to defend their conjunction of homology with common ancestry, whereas critics object that it confuses definition with explanation and leads to circular reasoning." The philosopher of biology Ronald Brady, one of the more outspoken critics of neo-Darwinism in the last generation, observed, "By making our explanation into the condition to be explained, we express not scientific hypothesis but belief. Dogmatic endeavors of this kind must eventually leave the realm of science." But the inference of common ancestry from homology had earlier run into an obstacle from an unexpected source, Gavin de Beer. A normally loyal Darwinian and embryologist, de Beer, 1899 to 1971, was a Fellow of the Royal Society. In the 1950s, he was the director of London's Natural History Museum, a successor to Richard Owen. In his 1971 book, Homology, An Unsolved Problem, de Beer wrote, "It is now clear that the pride with which it was assumed that the inheritance of homologous structures from a common ancestor explained homology was misplaced, for such inheritance cannot be ascribed to identity of genes. The attempt to find homologous genes, except in closely related species, has been given up as hopeless." It's surprising to find so well-established a figure as Sir Gavin, he was knighted in 1955, accusing Darwin of pride. But de Beer had identified a serious problem. Homologous structures need not be controlled by identical genes, he wrote, and homology of phenotypes does not imply similarity of genotype. If inheritance is the key to homology, as Darwin maintained, and what is transmitted throughout the generations is DNA or genes, then homology should be correlated with genes. But tracking the relationship between genes and bodily form, or morphology, has turned out to be difficult, as Harvard's R.C. Lewontin pointed out years ago. He wrote in The Genetic Basis of Evolutionary Change, 1974, that we do not know how to incorporate gene changes into a theory of speciation, and that in large part that is because we know virtually nothing about the genetic changes that occur in species formation. His comments were as illuminating as they were radical. Mayer had mentioned a genetic revolution, Lewontin wrote, but all such talk had to be held in abeyance until we could characterize the genetic differences between the populations at various stages of phenotypic divergence. The whole concept of a genetic revolution was driven by the idea that every gene affects every character, Lewontin went on. But it does not follow that every gene substitution really matters. It may be true that thou canst not stir a flower without troubling of a star. But, he continued, the computer program for guiding a space capsule does not, in fact, have to take my gardening into account. The problem of making quantitative statements about the multiplication of species has been that we have been unable to connect the phenotypic differentiation between populations, races, semi-species, and species with particular genetic changes. Put another way, evolution by the mutation of DNA is one thing, and the development of new phenotypes is another. And we still don't know how the two are connected. What we do know is that they are not connected in the simple, straightforward way that Darwinism calls for. It gets worse, because structures that are not homologous are sometimes produced by organisms with similar genes, while, as Michael Denton pointed out, Apparently homologous structures are specified by quite different genes in different organisms. Homology and embryology. Efforts to correlate homology with embryonic pathways have been no more successful than they were with genes. Gavin de Beer went further and questioned whether embryology sheds any light on homology. But to re-examine the issue, we must once again turn back to Charles Darwin. He introduced Ernst Haeckel into the story, writing in the 6th edition of The Origin of Species, 1872. Professor Haeckel, in his Generale Morphologie and in other works, has recently brought his great knowledge and abilities to bear on what he calls phylogeny, or the lines of descent of all organic beings. In drawing up the several series, he trusts chiefly to embryological characters, but receives aid from homologous and rudimentary organs. He has thus boldly made a great beginning. A German biologist and a great Darwin admirer, Haeckel, 1836 to 1919, for a while persuaded much of the biological fraternity that embryology was the key to understanding community of descent. He did so with his drawings of the embryos of different vertebrate classes. Critics today call the drawings deceptive or even faked, but his slogan nonetheless became famous. Ontogeny recapitulates phylogeny. Ontogeny was Haeckel's word for the development of an organism in an embryo. So what the slogan meant was that the successive stages of an embryo would retrace the evolutionary stages of its early ancestors. But Haeckel misled his viewers by omitting the early embryonic stages entirely, when the embryos of different groups look quite different, and then focusing on the middle stages, when they do become a little more similar. But he redrew them to make them look more similar than they really are, and that was where the deception came in. For this reason, his drawings have been discredited. Nonetheless, he did persuade Darwin, who wrote that the leading facts in embryology, which are second to none in importance, are explained on the principle of variations in the many descendants from some one ancient progenitor. He also wrote that early embryos show us more or less completely the condition of the progenitor of the whole group in its adult state. In a letter written to Asa Gray in 1860, Darwin considered embryology to be by far the strongest single class of facts in favor of his theory of descent with modification. Haeckel's embryo drawings became a leading icon of evolution in Jonathan Wells's book of that name. Wells also pointed out that Douglas Futuyma's textbook, Evolutionary Biology, says that early in development, human embryos are almost indistinguishable from those of fishes, while Brown University biologist Kenneth Miller described the development of the human embryo as a microscopic trip through evolutionary time. But the embryologist Gavin de Beer was highly critical of such claims. It does not seem to matter where in the egg or the embryo the living substance out of which organisms are formed comes from, he wrote. Correspondence between homologous structures cannot be pressed back to similarity of position of the cells of the embryo or the parts of the egg out of which these structures are ultimately differentiated. De Beer also asked what mechanism could result in the production of the same patterns in spite of their not being controlled by the same genes. I asked this question in 1938, he added, and, as of 1971, it has not been answered. It still has not been answered today. Automobiles in embryo. A fundamental feature of the evolutionist approach to homology is that it cursorily dismisses all design-based explanations. Darwinists long ago abandoned any thought of such a solution to the problem of homology, or to anything else in biology. In fact, Darwinism from the outset was intended to supplant design. But we are under no obligation to follow that example. Today, surrounded as we are by designed objects, it is not hard to see that homology flourishes in the world of design. The best-known example of evolutionist confusion on this issue was provided by the biologist Tim Berra of Ohio State University. In his book Evolution and the Myth of Creationism, Stanford University Press, 1990, he wrote, "If you look at a 1953 Corvette and compare it to the latest model, only the most general resemblances are evident. But if you compare a 1953 and a 1954 Corvette side by side, then a 1954 and a 1955 model, and so on, the descent with modification is overwhelmingly obvious. This is what paleontologists do with fossils, and the evidence is so solid and comprehensive that it cannot be denied by reasonable people." Philip E. Johnson of UC Berkeley referred to this as Berra's blunder. Corvettes of whatever year were all designed, and that indeed is overwhelmingly obvious. Yet Berra was so much enthralled to Darwinism that he even referred to year-over-year design changes as descent with modification. He had forgotten that a naturalistic explanation of homology must exclude design, not appeal to it. We might pursue the automobile metaphor further and briefly consider an auto assembly line, which is analogous to embryo development in biology. The completed car emerges at the end of the line. What's interesting about real assembly lines, visitors can see one in operation at Ford's River Rouge plant in Dearborn, Michigan, is the complexity of the operation compared to the finished vehicle. Machines do the heavy lifting, but the line is monitored by humans. At the first sign of a glitch, they can halt the whole operation. The assembly line is repeatedly updated to incorporate new models. Its planning and the importation of various auto parts are all off-stage and cannot be detected by studying the completed vehicle. Today, that vehicle is likely to incorporate parts that come from different parts of the world. Yet it is also replete with parts that are homologous with structures in vehicles made by different companies, some made in Germany, some in Japan, and so on. To cite only the most obvious homology, almost all such cars are quadruped, or four-wheeled, but there are many other such homologies. Michael Denton concluded, "A convincing explanation for the mystifying unity of type, the phenomenon of homology that Darwin thought he had so adequately explained by descent from a common ancestor, is probably still a very long way away. With the demise of any sort of straightforward explanation of homology, one of the major pillars of evolution theory has become so weakened that its value as evidence for evolution is greatly diminished." In The Living Stream, 1965, the British biologist Sir Alister Hardy said that the concept of homology is absolutely fundamental to what we are talking about when we speak of evolution. Yet in truth, we cannot explain it at all in terms of present-day biological theory. One is inclined to say that if homology is the best evidence for Darwin's theory, as some have thought, then that theory doesn't have much to recommend it. Further, that evidence is plainly more compatible with design theory than with descent with modification. In the next chapter, I shall review the evidence for convergence, the undoubted similarity of traits in organisms that no one supposes to share a common ancestor.